Thank you. Can you all hear me if I speak in this kind of voice? Yeah? Yeah. All right. So thank you for inviting me. I've never been here before. I have a strong connection to Spain because my wife is Spanish from El Puerto de Santa María. Nuestra hija nació en Jerez. And I also did a postdoc in Madrid about 10 years ago. And I know Carlos and Jennifer since a long time ago, since, I don't know, 15 years. Okay, so I'm going to talk about, the title is Using Palaeogenomes to Explore the Evolutionary History of Pleistocene Megafauna. And before I get into the data and the analysis, I'm going to start with acknowledging some people. So I'm doing it kind of reversed. But I really want to highlight the people who are behind. I'm essentially talking about two papers that were published last year. And the key persons in this is L. Palkopoulou, who was my PhD student, and Pontus Skoglund, who used to be a PhD student in Uppsala, and he's now in Boston, in Harvard, with David Reich, who's here. This is Sergei Vartanyan, who's our Russian collaborator. This is Henrik Poinar, and this is Anders Götterström, who's in Uppsala. And these are really the key players without whom, especially L and Pontus, this project would never have happened. So what I'm going to talk about is, first of all, I'm just going to give you a very brief introduction to ancient DNA and the type of sampling that we do, and very quickly in one slide, just how we do the analysis. And I assume for most of you this is old news, but I'll go through it quickly. Then I'm going to talk about how we can use ancient or prehistoric genomes to look at population divergence in the first part, and that's going to be about half the seminar. And then in the second part, I'm going to talk about how we can use ancient genomes to reconstruct demographic histories and also to look at genetic stochasticity. Right. So to start off, just the general environment that we typically get our samples from is this. This is the high latitude northern permafrost tundra. This particular picture is from the Taimyr Peninsula in northernmost Russia, about, I think, 75 degrees north or 70-something degrees north. And as you can see, it's really flat, it's really wet, and what you can't see is that it's most of the year is very cold because it's very far north. And you have lots of extant animals that live in the north. These are Norwegian lemmings that we do a lot of work on as well, and Arctic foxes that I did my PhD on. So these species all live there today. But today I'm going to talk about mostly ancient material from long gone animals. But I wanted to mention before I do that, and I was supposed to play a movie, but it didn't work. But I have a picture instead. So the most common animal up here, which is a hassle, are the mosquitoes. Someone really should do a project on these. These are horrible, and they are there all the time. This photo was taken in July last year in northeastern Chukotka. This is my colleague from the museum in Stockholm, trying to drink water with mosquitoes inside. Anyway, so Siberia typically looks like this. It's fairly flat. You have all the modern day animals living on top of the tundra. But what I'm going to talk about are samples that are buried in permafrost. Okay, so all this is permafrost. It's essentially frozen mud with a very high organic content. And what happens if you go to Siberia or to Alaska is that you typically find along either the ocean, as in this case, or along a river, or along a lake, you find these sections where the water every year is eroding the permafrost. So you get these cliffs here. And inside this kind of, in this case, I guess it's about 10 to 15 meters thick here, inside this frozen permafrost, you have lots of bones from dead animals. And as the water is eroding this kind of cliff, these bones fall out. And so to find them, you essentially go beach walking or mud walking, as in this case, and you look for bones. It typically looks like this. This is the same beach that we just saw. Here is a scapula, so a shoulder bone from I think it's a bison, a steppe bison, an extinct species, one could argue, from the Pleistocene. And this is often quite dirty work. You normally get really muddy, and it's like being a kid again. It's fantastic. This is me. This is femur, I think, from a woolly mammoth, and this is bone from a woolly rhinoceros. So what we do is that we collect samples. We have, of course, have collaborators that collect samples in the field, and we bring these dirty bones into our ultra-clean ancient DNA lab. We try to clean them as much as possible, and then it looks like this. So we wear silly spacesuits, and this is to protect the samples from our DNA. And what we typically do, this is a K-bear, this is a bison, is that we remove the surface of whatever bone we want to sample using sandpaper or a drill. And then we take a small piece of the bone or tooth from inside and in the hope that this will be fairly clean. So it won't have lots of environmental DNA from soil microorganisms or from modern humans and so on. So we try to keep this as sterile as possible. Then generally what we do is that we extract the DNA using standard methods, EDTA to demineralize the bone, proteinase K to dissolve the proteins. And we use urea, which helps the extraction process. And we typically filter these, the extracts, and then we purify it using silica and various buffers. Then most often we nowadays build sequencing libraries using standard method, Meyer and Kircher methods, so single index libraries. And normally when we have a lot of samples, we then index these using an indexing PCR so that we can pool many samples onto one run for screening purposes. And the reason we do this is that most of these bones, they have a very small amount of endogenous target DNA from the animal that you're trying to analyze. So it might vary from anywhere between below 1% of the DNA, that is your organism in question, up to perhaps 85, 90% is the highest we've seen. And obviously, if you're trying to sequence something like a Neanderthal, like the first Neanderthal paper, where they had about 1% endogenous DNA, you're going to have to spend a lot of money sequencing those 99% bacterial DNAs that are in there. So you don't want to, if you can choose, you want to find the samples. You want to identify the samples that have a very high endogenous DNA content. Once we do that, we then try to sequence more deeply to get complete genome sequences. Okay, so that was a very brief introduction. Now I'm going to talk about the first part, about population divergence. And we are now, this is kind of a story that I'm going to tell you. And we'll see if you believe it or not. So this is a picture taken on the Taimyr Peninsula in 2001 in July. And we were here collecting bones, as I showed you before. And along this little section over here, we did find a bone that was a small bone, it was broken, and I couldn't identify it. I thought it was a reindeer. I just have to admit that. Okay? I had no idea what it was. I wrote reindeer on the bag, but no clue. Here's a photo of this bone. It's this bone. So I used a small saw, a Dremel tool, to subsample the small piece, this one. Put it in a bag, brought it back to Stockholm. These other bones are, these two are mammoth. This is a bone, or part of a pelvis. This is a tusk from a woolly mammoth, and so on. This is probably a bison, and this is a horse, I think. Okay, so once we got back to Stockholm, we used 16S, which is it's a short part of the mitochondrial genome. We amplified that and sequenced it for all these specimens that we couldn't identify. And the idea here is that this is a very good diagnostic marker for species level relationships. Once we did this on this is a little bone piece, it turned out to be a wolf. So in some ways that's a disappointment because Taimyr, northern Russia, is full of wolves today. So we thought this bone was probably going to be maybe 5 years old or 10 years old or 100 years old, but probably not very old because it's out of context. It was just lying there on the beach. But then we radiocarbon dated it and it turned out to be 35,000 years old. So that's interesting. We now have a wolf bone that's 35,000 years old. And we decided to use these methods that I just talked about to sequence it, and we managed, I think the endogenous DNA content on this one was all right. It was about 15, 16%, but that was okay. So we managed to sequence the complete nuclear genome at 1x coverage from this specimen. So then the question is, what can this 35,000-year-old wolf genome tell us about canid history? Well, it might tell something about the origins of dogs. It might not. as you probably all know, there's been a huge amount of papers published on where and when dogs were domesticated. And many of these papers have been co-authored or authored by Jennifer and Carlos. So, for example, this is a paper where Jennifer is involved, and this, I think, Carlos is there somewhere. Now, I'm not going to review all these papers. I'm just going to perhaps mention that there is a slight disagreement in the scientific community about when and where dogs were domesticated, especially where, but also a bit about when. Okay, so if we focus on, I should go back. If we focus on this paper, which at the time when we did our study was probably the best paper out there looking at the origin of dogs, so the relationship between dogs and wolves, the Friedman et al. Paper. Essentially, very briefly, what they did was that they sequenced complete genomes from multiple dogs and wolves. They used modeling to try to estimate the divergence time between dogs and wolves, assuming that this divergence probably represented the timing of domestication. Interestingly, it turned out that wolves here are somehow in this tree monophyletic, which, at least to me when I read this paper, was a big surprise. You would have thought that the wolves would have this huge tree and then dogs would be nested within this. That's what I would have expected, at least. Anyway, when Friedman and colleagues estimated the split time between dogs and wolves, they found that if they assumed the mutation rate of 1 × 10⁻⁸, the split time was sometime around 11 to 16,000 years ago. And it should be said, since people disagree in this business, that other people have published other papers assuming other mutation rates and thereby coming up with different estimates, ranging from, I think, 10,000 in the Axelsson paper up to 30,000 in Jennifer's paper and also the Wang paper that is cited here. So when we have a 35,000-year-old wolf genome and we're looking at this paper, our expectation is that our wolf is going to be up here, right, 35,000 years ago. So is that really going to tell us anything about the origin of dogs or the split between dogs and wolves? It might not. At any rate, this wolf should have lived a long time before the inferred split between these two taxa. Okay, I have a watch here on my, oh, there we go. Okay, sorry, I'm a bit confused. Right, so what we decided to do, first of all, since we have a very clear null hypothesis, is that if you look at wolves and dogs, and a split that in the Friedman paper should be about 10 to 15,000 years ago, we expect our wolf to be up here somewhere. So therefore, we predicted that this wolf would be symmetrically related to all modern-day dogs and wolves. So we can test this using what is famously called the Abba-Baba test, or the D-statistic. And when we do this, basically you mine your genome sequences for sites that are polymorphic, where your outgroup has one variant, and then your donor population has the other SNP, and then your two potential recipient populations are variable, or they differ from each other. And if you have no particular relationship between this donor taxon and either of these recipient taxa, then you expect an equal number of trees looking like this and like this. That's essentially the D-statistic. So using this formula, then your expectation is a D-statistic of 0. So when we test this, we find that indeed the Taimyr wolf in any constellation, when we compare to these modern genomes, has a D-statistic that is, we can't separate it from 0. Okay. So indeed, the Taimyr wolf is symmetrically related to modern-day wolves and modern-day dogs. Fine, that was our null hypothesis. But then we also used, or Pontus, I should say, used something called admixture graph models, where you can try to reconstruct the relationship between different taxa and different nodes. And you do this by way of trying to minimize the amount of genetic drift that has occurred between the different taxa and the different nodes. And when Pontus did this, the Taimyr wolf ends up here, not really like a trifurcation, but at any rate non-significantly differentiated from the node. So that was a bit odd to us, right? The Taimyr wolf we expect to be up here, fairly significantly differentiated from the common ancestor between wolves and dogs turned out to be non-significant. But maybe that's just a power issue. Who knows? So how do we address this? And what Pontus suggested to do was to then use simulations to try to address this using something called FAB, which is a statistic where we estimate the probability, I have to almost read this, that the Taimyr wolf shares a derived allele discovered as a heterozygote in the Chinese wolf genome. We chose the Chinese wolf because it seemed to be the best genome, the highest coverage, and also cleanest in terms of past introgression with dogs and so on. And we also excluded CPG sites, and these are sites in ancient genomes that are difficult to get rid of damaged bases from. Okay, so our libraries had been treated with something called the user enzyme that cuts up all these nucleotide bases that are damaged, but it's difficult to get at these CPG sites, so we excluded those. And we then ran simulations of the divergence time between the Taimyr wolf and the Chinese wolf, assuming a series of different mutation rates. And then we used the FAB statistic as a summary statistic in these simulations to try to infer the mutation rate. Okay, this is what the results look like. All these different colored lines are the different simulations. Here's the FAB statistic. This is the population split time between the Taimyr wolf and the Chinese wolf. And our estimated FAB statistic, the summary statistic, is here. And then we also know the radiocarbon age of the Taimyr specimen. And so there must be at least 35,000 years of evolution between the Chinese wolf and the 35,000-year-old Taimyr wolf. There could be more if there is structure in the data, but at least we get a maximum mutation rate out of this. And that occurs where this summary statistic intersects with the simulation and our radiocarbon date. So 0.4 by the power minus 8. So if you remember when I talked about earlier the assumed mutation rate in the Friedman paper, that was 1 by the power minus 8. So our inferred mutation rate in these canids is about half of that. So when we then recalculate the split time in the Friedman paper using this mutation rate, we infer a split between modern wolves and modern dogs at around 27 to 40,000 years ago. So we have two, not lines of evidence because it's the same genome, but we have two different kinds of analysis. One is a mixture graph saying that this Taimyr wolf seems to be not significantly differentiated from the common ancestor of modern day dogs and modern day wolves. And secondly, we have this when we calibrate the mutation rate and we find a very similar answer, so 27 to 40,000 years ago. So both these analyses seem to suggest that modern day wolves and modern day dogs diverged around 35,000 years ago. There was a paper that came out by Adam Boyko's group just before Christmas in PNAS that used a huge amount, thousands I think, of dogs and wolves, but SNP data. And Pontus immediately extracted all that data and built a tree where he included the Taimyr wolf. And this is a bit messy. All this is dogs. And for some reason, all the present day gray wolves are on this little branch. And here's a Taimyr wolf. So what I wanted to show with this is just that, again, in this tree, the node here, where the Taimyr wolf and the split between wolves and dogs, they're very close together in time. So again, we get support for the idea that the divergence happened around this time. Okay, I'm going to just briefly leave the dog business and mention one other result in this paper. Is that we also did exactly the same kind of test, the D-statistic test, using the Taimyr wolf against all modern day wolves. And we didn't have very many genomes because at that time there wasn't any published. But there was a huge data set from Birgit von Holt and colleagues on SNP data from many different wolf populations. And when we do the statistics again, so you can ignore this one, this is more like a control that we have power. But this is the D-statistic where the Taimyr wolf is the donor population, and then you have pairs of wolf populations that represent the potential recipients. These are all at the 0, so they are all non-significant from 0. And again, that means that it looks like the Taimyr wolf was also symmetrically related to all modern day wolves, including modern day wolves, and you can't really see that here in North America. So this would indicate that sometime after 35,000 years ago, all modern-day wolves might have shared a common ancestor. And that in turn could indicate that there might have been a bottleneck in the worldwide wolf population sometime after 35,000 years ago, presumably before 12,000 years ago, because that's when the Bering Strait, the sea levels rose and North American wolves became isolated. So not really hard data on this because it's based on a comparison of an ancient genome data and SNP data, but nonetheless it's an interesting observation. Okay, so to summarize part one, we infer that the split between dogs and wolves, the modern day dogs and wolves, probably happened sometime between 27 and 40,000 years ago. This could either represent the domestication, the start of the domestication process, but it could also mean that there was a major split in the wolf population at this time, or one of these wolf lineages led to all modern day wolves, and the other wolf lineage led to modern day dogs. But then those wolves that were on that branch, they must then have gone extinct in the wild. Otherwise, we wouldn't get the result we did. So just to show this in a very easy sketch, either you have a split here that represents the domestication of 35-ish, 27 to 40,000 years ago, or you have a split in the wolf population, and the domestication starts later, but then this wolf population is extinct. So I'm not going to argue which one is true. I just think that this one, to me, is more parsimonious, but not by much. Okay. And then finally, there's also an indication that wild wolves might have gone through a major bottleneck sometime between 35 and 12,000 years ago. All right, so that ends my first half of this presentation about population divergence. And I'm going to stop talking about wolves now. Is that okay? Yeah? You don't want to hear about wolves anymore? Okay. So, now I'm going to talk about demography and genetic stochasticity. So genetic stochasticity, as I'm sure most of you are aware, is a term that encompasses both genetic drift and inbreeding. And I'm going to start very, very simple. So imagine any endangered species, more or less. It will have a small population size today, and sometime in the past, this population or this species had a large population size. It's very simple. The same goes for extinct species, of course. So at some point in time, you have a demographic decline. Okay, so this could lead to a number of different, this is very basic, I know, number of different problems, right? You could lose genetic variation, thereby losing adaptive potential. You could get inbreeding in the population, leading to expression of recessive deleterious alleles, et cetera, et cetera, et cetera. This is a quote from Ian Barnes, who's a funny guy. I like that. Anyway, okay. So you would think that the IUCN used genetic parameters as one of the criteria for classifying the red list, but they don't. In fact, if you read the whole document about red listing criteria, you barely find, you cannot find the word genetic in there. I think it's mentioned once and only like in the passing. So why is that? I'm sure there are many political reasons for this and so on and so forth, but one reason is that up until recently, we haven't been very good at measuring the whole genome-wide diversity in species. We have been using mitochondrial DNA, we have been using a limited number of microsatellite markers, maybe 10, maybe 15, and then there's been a bunch of papers arguing, mainly by French people, arguing that that's not nearly enough to measure the whole genome-wide diversity. So maybe that's one reason. But then again, nowadays, in the last 5 years, people have started to sequence complete genomes from a bunch of different endangered populations and species. So why aren't we using it now as a criterion for just, here's a measure of the genome-wide diversity. Let's just take that and use it as information for whether something should be red listed as only vulnerable or critically endangered. Well, one additional reason for that is that there's actually, if you compare different species, there's actually no clear relationship between genome-wide diversity and how endangered a species is today, or how rare it is today. So here's a figure from a paper we published last year that L. Palkopoulou put together, which has a bunch of data from whole genomes from different endangered species and heterozygosity down here. If you look closely at this, you will see that there's no clear pattern where these ones are really rare and these ones are a bit more common. Instead, at the top here, you have the Sumatran orangutan. It's one of the most endangered species on the planet, and it has a fairly high diversity. Humans are somewhere in the middle here. Whereas at the bottom here, you have the polar bear. The polar bears are not endangered. They will be, but they're not endangered today. There are tens of thousands of polar bears around. They're nowhere near the Sumatran orangutans, and still they have a really low genome-wide diversity. And the reason for this is, of course, obvious. Right? One is that your genome-wide diversity is affected by ancient history. So the reason all humans outside Africa have a comparatively low diversity is because we went through a bottleneck when we left Africa. But another reason is also that different species will have different life histories, different mating systems, which affects the effective population size, perhaps different mutation rates, and different generation lengths. So all this affects it. So our argument, or my argument in this presentation, is going to be that it's not going to be very useful to run around sequencing a bunch of modern genomes and trying to use that as information for how threatened the species is. I'm sure people will disagree with this. But our argument then is that instead of doing this, you could take one particular species, you can use ancient DNA technology, and you can go back in time and you sample this species or population from before it started to decline, and then you sample it at present day. Right? So you sample across this declining population size, and then you can actually measure within the species how much genetic variation it has lost and how much inbreeding it has accumulated. So you don't have to compare it to other species. So what better species to do this on than an extinct species already, the woolly mammoth. So in order to kind of test this idea, we decided to look at the woolly mammoth. This figure here is a skyline plot done in BEAST to reconstruct the demographic history of the woolly mammoth. So you have effective population size on this axis, and you have time here. And this is based on mitochondrial DNA sequences from several hundred mammoths. And the reason I'm showing you this is just to show that woolly mammoths used to be, during the Pleistocene, during the last ice age, they used to be really, really common all over the place, all over northern Eurasia. And then at the end of the last ice age, according to the mitochondrial DNA, the population size crashed down to a very small level, or comparatively small level, and then it was kept there for a number of thousand years, and then mammoths went extinct. So the demographic history of the mammoth, going from very large to very small, and then being kept small for thousands of years before going extinct, is actually a pretty good model to test this on. So what we could do is to sample, for example, a mammoth here, when there were lots of mammoths, and we sample a mammoth here just before they became extinct. And we measure the genome-wide diversity and inbreeding in these two specimens. And before I get into the results, I'm just going to tell you a little bit about the history of the woolly mammoth. This is a map. It shows the eastern part of Siberia and Alaska. And all that is yellow here was dry land during the Pleistocene, because the sea levels were about 100 to 150 meters lower. And mammoths roamed all this area, and they also went all the way through Eurasia, all the way into the United Kingdom, down to Spain even, I think, down in close to Malaga, right? Almeria, yeah. Anyway, so there was lots of mammoths around. And what happened at the end of the Pleistocene, at the end of the Ice Age, was that mammoths became extinct everywhere on the mainland around the same time. 12,000 years ago, mammoths go whoop, gone everywhere, except on this little hill, because this was a hill when the sea levels were low. Because when sea levels rose at the end of the Pleistocene, so this became an island. This is Wrangel Island off the Siberian coast, and mammoths survived here from about 10,000 years ago, when all the other mammoths had gone extinct, until 4,000 years ago. And then they finally became extinct. And so two things. We could then sample. We had one sample that turned out to have really good DNA from here, and then one from Wrangel that was 4,300 years old, just before the extinction. And Wrangel Island is also a very small island. The carrying capacity of Wrangel Island, roughly, is between 200 and 1,000 woolly mammoths. So this population that survived here for thousands of years must have been fairly small. Okay, so after a lot of screening of lots of samples, we targeted these two particular samples that had really high endogenous DNA counts, so up towards 80% for one and 60% for the other one. We sequenced about 900 million reads, blah, blah, blah. We, in the end, got, using the African elephant genome as a reference sequence to map all the reads to, we recovered about 17x coverage of the Wrangel mammoth genome and 11x coverage of the Oymyakon, the old 45,000-year-old genome. This is L in our lab, okay, who did all the work on this paper. So what can we do with this? First of all, what most people do when you have a more than 10x coverage genome is to run a PSMC analysis. So reconstruction of the demographic history using a single diploid, fairly high coverage genome. So we did this. And so this is just a figure showing a heterozygosity across one of the chromosomes. And I'm sorry you can't read this, but it says 100 megabases here and about 200 megabases here. So the PSMC uses the amount of variation, which equates to the collection time, for different blocks across these chromosomes to estimate periods when the population size was small, essentially. When we do this for these two mammoths, the PSMC plots come out and look like this. Okay? So we have some problems here. It's a nice curve for both of them. They're fairly similar but different at the same time. But what we don't know is when things are happening. Down here it says time scaled in units of 2 multiplied by mutation rate by time. So in order to make sense of this, we need to know the mutation rate, right? Which we don't know. We could make a guess, but it would probably be the wrong guess. So what we decided to do was to use there are 2 points of information that we could use to perhaps calibrate the mutation rate in these genomes. One is that we know that the end point of this PSMC plot in the Oymyakon genome is 45,000 years old, right? This line ends at 45,000. This line ends at 4,000 when the Wrangel individual died. So there is about 40,000 years of evolution missing in the Oymyakon genome that we see in the Wrangel genome. That's one point. The other one is that we make an assumption and we say this bottleneck here is probably the same in both these species. And by doing this, and I'm going to show you graphically, and that's not exactly how it was done, but you can merge these. Okay? And since you know by how much you need to move these together, you can then infer the number of mutations that you had to move them together. Because essentially on the x-axis you have number of mutations. And from that, you then know the mutation rate. Okay? So using this approach, we could then estimate the mutation rate across genomes using just the fact that we know that they are 40,000 years apart. And the estimate we get is 1 by the power of minus 9 mutations per base pair per year, which is a high mutation rate. It's higher than humans, but twice humans, actually, the most recent estimates in humans. But it's the best one we have, we argue. When we then do this, use this mutation rate, we can then convert this time axis into years up here. And then we can infer 2 things from this plot. One is that this bottleneck that seems to have been very severe appears to have happened sometime around 300,000 years ago. And we have no idea why. It just doesn't make sense. So it's a mystery for the future. I thought that we would find a bottleneck 120,000 years ago, the last time the climate was really warm and Arctic species in general were not very happy about that warm climate. But no, it's older than that. We don't know. Maybe there is something wrong in the PSMC. I don't know. But we don't know why this happened. If we try to plot this on the climate curve, there are several interglacials happening and ice ages happening during this wide time window. So we simply don't know. The second thing that is noticeable here is that from around here to here, which in this time scale is from 20,000 up to 10,000 years ago, there is a severe decline. And that, of course, fits with what we know about the history of the woolly mammoth going from a very large population to a very small population. And then the last about 8,000 years of its existence, or 6,000 years, there are very, very few mammoths left. Okay. So what about genome-wide diversity? When we measure this for these 2 specimens, we find that in the Wrangel genome we have about 1 heterozygous site per 1,000 base pairs, whereas in the Oymyakon genome we have 1.25. So in fact there is a 20% difference in genome-wide diversity between these 2 specimens. So we can kind of naively fill this in. The delta H is 20%, a 20% loss in diversity, based on 2 samples only, I know, but still. So what about inbreeding? Well, you can use, as again, I'm sure most of you already know, you can use whole genomes to estimate the genomic inbreeding in an individual. The way this works is that you look at diversity across the chromosome. This is the Oymyakon genome. You can see it's kind of variable all across. It differs from different parts of the chromosome or the genome, but it's fairly variable everywhere. The Wrangel genome doesn't look like that. The Wrangel genome has huge, or not huge, but it has big chunks of no variation at all, completely homozygous. And these ones are around 1 to 2 megabases. This one is about 15 megabases long. This is strikingly different from the Oymyakon genome that is variable. And this is just an example of a small part of one chromosome, but it looks like this across all chromosomes. So what we could do, for example, is to just count how many of these blocks do we have that are bigger than 500 megabases, than half a megabase, compared in the Wrangel genome compared to the Oymyakon genome. And we put this into a table, and we find that actually the green here is the Wrangel genome. So here you have the blocks, the runs of homozygosity in megabases, and here the frequency of those. And you can see that the green Wrangel has a very high frequency of runs of homozygosity, whereas the Oymyakon genome has just a few. You have to zoom in actually to see that it's about 10 across the whole genome, so very few. To convert this into numbers, there's a 28-fold higher frequency of runs of homozygosity in the Wrangel genome compared to the Oymyakon genome. And overall in the Wrangel genome, or I can start with the Oymyakon genome, had less than 1% of its genome was in this state of homozygosity. In contrast, the Wrangel genome had 23% of its genome in runs of homozygosity. So this is roughly equivalent to an inbreeding coefficient of 23%. So nearly the result, one would think, of full SIB mating. But what's interesting here is that for full SIB mating, you would expect runs of homozygosity that were tens of megabases long, or even longer. That's just how genetics works. But the Wrangel individuals' runs of homozygosity are, almost all of them are less than 2 megabases long. So what we infer from this is that this Wrangel individual, its parents, his parents, it was a boy, his parents were not siblings. That's impossible given our genome data. Instead, it looks like the genome in the Wrangel mammoth is a consequence of multiple generations of matings between distant relatives. That would kind of fit this pattern. There might be other explanations involving ancient bottlenecks and recombination, but these are, in my mind, less likely at least. So this is exactly what you would expect in a very small population that is randomly mating, probably even displaying inbreeding avoidance, but then just by random chance, you end up mating sometimes with a second-order cousin that you didn't even know was your cousin. And there you go, for thousands of years, you will accumulate small blocks of inbreeding in the genome. So we can then fill this one in if you want. Delta F is 23 or 23%. And I would just like to stress that, of course, we have no idea whether this inbreeding that we infer had any effect on fitness. Okay? We, as of yet, have not been able to test this. We are working on different ways of doing this, but it might be that this had no effect on the population's fitness. On the other hand, if we look at extant populations that are inbred, quite often you do find a negative impact on fitness in these review papers that have been done on this. So it seems perhaps probable or likely that it had some sort of effect on the fitness of the Wrangel mammoth. Maybe it had made some sort of contribution to why the mammoth became extinct, even though there's nothing to say that it was a driving force behind it, or a major force. So to summarize the mammoth story, part 2, we infer a demographic bottleneck in the middle Pleistocene 300,000 years ago that we don't know what it's about, and we also find a very sharp decline in population size around 20 to 10,000 years ago at the end of the last ice age. In this last surviving mammoths, just before they became extinct, we do find some evidence that they have lost diversity, and we find some evidence that they have accumulated inbreeding up to 20%, 23%. And which I didn't show you, but we can also use this genome data to infer the effective population size on Wrangel Island during these last 6,000 years. And our mean estimate is about 240 effective mammoths. So this is well below this magical 500 number in conservation genetics, which you according to Russell Landy and colleagues, should try to stay, Frankham and colleagues, should try to stay above in order to not gradually lose genetic diversity. So in order to preserve your adaptive potential, if something in the environment changes, you want your population above 500 in NE. And it appears that the mammoths were below that, so maybe that is the reason why they had lost genetic diversity. So, I have plenty of time. I only have one slide left anyway. Okay, so I'm just very briefly going to tell you about our future plans. So what we want to do is, first of all, we only have 2 genomes from the woolly mammoth right now. And there's 40,000 years between them. So we don't really know when the diversity started to decline. We think it happened at the end of the Ice Age. But of course, we're trying to get many more genomes from mammoths to really pinpoint when things are changing through time. So right now we have about 150 samples that are dated to the Holocene from Wrangel Island. And we have another about 100 to 200 samples from Chukotka Peninsula, so the ancestral population before the sea levels rose. So hopefully we're screening these and trying to find samples that have a high endogenous DNA content, and hopefully, assuming we get some more money, we can sequence many more mammoth genomes and really pinpoint what is going on here. But what we also want to do with the data we have and data we are getting soon is that we want to look at a number of different parameters that I think could give us an indication whether this loss of diversity is also leading to genetic problems in the population. So one way to do this is to look at the DNDS ratio, so the ratio between non-synonymous and synonymous mutations in the genome. Because normally in a large population you expect natural selection to purge, to remove these non-synonymous changes as they happen. You have a sort of balance between drift and selection there. But then in a small population where drift becomes stronger, this balance point changes, or in theory it should change. So you would expect natural selection to be less good at removing these deleterious alleles. Then there are certain conditions in small populations where actually maybe selection is able to purge these deleterious variants as well. And you see this in many populations or species that are the result of very strong founder effects. For example, in Hawaii, etc., you find these elevated DNDS ratios, and presumably this is because of this bottleneck effect when they founded these islands. So we want to test this. We want to look at how the DNDS ratio changes over time, whether we can find any evidence of either an increased fixation of deleterious alleles that are recessive, or whether we might find the opposite. We might find that actually these recessive deleterious alleles might actually be purged from the population through time, through natural selection. So it's an interesting kind of set of hypotheses that hopefully we can test. And of course we are also, and we have already done this actually, but I don't have a slide on it, but we can also look at diversity in parts of the genome that we expect have their variation maintained by balancing selection. So the classic example is the MHC locus, that normally even in the Channel Island foxes that have no variation anywhere, they appear to have maintained MHC diversity because of balancing selection. And we've done this, I don't have a slide on it, but it looks like MHC diversity is actually lost in the mammoths on Wrangel as well compared to the ancestral population. Not by much, but it's lost at the same rate as it's lost at about 20 to 30%, so the same rate as we find across the genome in general and also in previous studies using microsatellites. And these types of analysis right now, we have started doing on other species than only woolly mammoths. So we are right now working on, apart from mammoth, we are starting a project on woolly rhinoceros, the same thing. The woolly rhino is also extinct. It has a very different history because it didn't go through this very long period of isolation on a small island. Instead, it just gradually became less and less abundant until it's disappeared around the end of the last ice age, 12,000 years ago. We're also started a project to do this on Sumatran rhinoceros using museum samples and modern samples. And we have a Sumatran rhino reference genome already, so we're now collecting historical museum samples. And the same thing for, we have 2 projects going on eastern gorillas, so Grauer's gorilla and the mountain gorillas, where we again are analyzing genome data from historical museum samples. And also we, together with Karin Norén, that Carlos knows, we got a grant to do the same thing on the Scandinavian Arctic foxes that have gone through a major bottleneck in about 100 years ago, where they went from 10,000 individuals down to 100, and they've been at that population size since then. So the idea is to do this on many different species to try to get a sense of how genetic variation and inbreeding is changing through time. Okay, and with that, I thank you for listening. Thank you.